Andrew, it's a great pleasure to welcome you to Liverpool, and please tell us all about Lost in Translation. Thank you.
Thank you, Chris, and to the Pain Research Relief Foundation. It's a huge honour. You're totally humble when you see the list outside of previous speakers here. And you've really put the icing on the cake by having the theme to remember the work of Ulf Lindblom, who was one of the greats. He was a predecessor of mine as president of ISP. He was influential to all of our careers, for instance, and his work carries on. So currently at Imperial College, we've observed that you can detect leprosy early using thermal sensory thresholds and diabetic neuropathy. And we're currently building low cost with our engineers thermal threshold devices. He was one of the greats, and I had the pleasure of meeting him very early in my career. I was taking on a challenging subject, which is challenging microneurography, and he was very supportive. The title is Lost in Translation. This is a bit of a private thing between me and Per, but I was a bit worried he might be an imposter, because I recognize the importance of ideas in your sauna. I have a Finnish sauna at home in Buckinghamshire, but he can't be a Swede, because my mother was Swedish and we would call it a bastu, not a sauna. You don't use these terrible Finnish words. It's like the Finns claiming that Santa Claus lives in Finland, which we know is complete. What I want to talk is about a lot of the work, and some of this is going to be challenging, around the failures in translation and developing new drugs. And what I don't want to do is people to go away with the impression that this is because I'm going to focus on animal models, because that's the area we've worked on, that this is a diatribe against the use of animal models. Absolutely it is not. But we have to be a lot smarter in the way we use them, and we have to ask much smarter questions of them. So these are some of my declarations, none are particularly relevant. But the last one is that I spend a lot of my career working with animal models, and I've made pretty much all of the mistakes I'm going to talk about. But we can't carry on doing things this way because it's simply not working. When Chris was kind enough to ask me to give this talk, we talked about a number of topics that I've done work on, and this was the least clinical of them. We could have talked about many other areas. And I didn't get his wisdom initially. I now do, because I've been thinking about this. So why am I talking about these translational problems to essentially, most of you, I suspect, will be from a clinical background. The truth is it's everybody's problem. Unless we get this right, we're not going to get new drugs and things like that. But it's also, I don't want to focus on the problems. I want to give you some of the solutions that we've developed over the years. And I am going to focus on animal models because that's what we've worked on. But in fact, most of the concepts I'm going to talk about are generic across the whole of pain research. And we really do need to get part of our house in order. And I'm going to give you tools. So if you're not a basic science researcher, these tools will still be useful. I was really lucky to be working in Oxford when Henry McQuay and Andrew Moore, I think both of them have been speakers here, but certainly Henry McQuay has, when they were starting evidence-based medicine. And they taught me one thing, because they said, which part of the paper do you read first? And it's, oh, the results or the bottom of the abstract series. No, you read the methods first. So I'm hoping to give you some tools whereby you can judge the rigor of research and read the method section first. I work a lot on leprosy, and I had the privilege of giving a plenary lecture at the World Leprosy Meeting in Manila a few years ago. And I was going to stick up a slide here that was outside the main auditorium in the big convention center just before I gave my lecture, which says no firearms allowed. But I resisted that, but please don't shoot the messenger, because this is an issue for all of us. So let's start with the uncomfortable truths. I've been part of a team that has published the NUPSIG revised guidelines. We published them a few weeks ago in Lancet Neurology. And I wasn't even comfortable talking about first or second line drugs with some of these areas. We're looking, and there are some we recommended against. But these are what we decided as first, second, or third line. But here are some of the truths. These are really modest efficacies. If you had an antibiotic that treated 1 in 9 people successfully, you would not very embarrassed. But that is where the alpha-2 ligands are, so gabapentin and pregabalin. fantastically well marketed. So what we're saying at the moment is most treatments don't work for most patients, but some, as good clinicians, we know that some treatments work for some patients. We just don't know who. And I think a lot of the problem with, particularly in the area of drug development, is we've been too generic. We haven't asked specific questions. We want the whole cake rather than taking a slice of it. And of course, this is at the basis of precision medicine. Ulf Lindblom was a sort of pioneer of that because he recognizes that there were differences in sensory profiles between different patients, and that's one of the areas that's being actively pursued. And next year's ISP Global Year, we haven't announced it yet, but it will be on neuropathic pain with a particular area around precision medicine and improving our research methods. And we've invested billions and billions in preclinical pain research, and it's taught us a lot about mechanisms. And a lot of us celebrated, was it 3 or 4 years ago, when David Julius and Ardem Patapoutian won the Nobel Prize, absolutely deservedly, for elucidating sensory transduction mechanisms. But at best, capsaicin works in 1 in 13 patients. Now, we actually used it in our clinical, and the patients who do respond well. In fact, during COVID, we recognized that we lost control a lot of them because they weren't coming back for their regular treatments. But the bottom line is we haven't translated these things into effective drugs. And there are many, many reasons for that. I'm going to give you one example. As Chris said, I had the pleasure of chairing the cannabis task force for ISP, and we published all the papers in one. And we came to the conclusion that cannabinoids were not effective for the management of pain, and they are not safe either. And that's controversial, but we really drilled down into the evidence, and that hasn't changed. But one of the things we did was a preclinical systematic review, and we identified 370 odd studies that were in the field. And if you look at them either for rats or mice, most of the reports show some efficacy of the ligands that were used. But there's a problem here. If you look at the reporting quality for the main risk of bias items, which is using a tool developed by our friends Camaraderies up in Edinburgh, you'll see that there's areas of red. And then if you use the circle tool, which looks at risk of bias as opposed to the reporting standards, you'll see it's mainly yellow, which means there's a lot of uncertainty about the rigor of this evidence as a field. And cannabis is just one example. So if you go to the clinical trials, we then identified 31 studies. There's a few more now, but it hasn't changed. That's over 7,000 patients, mainly in neuropathic pain. So particularly if you can combine it with neuropathic pain after multiple sclerosis. Interestingly, there's a big disconnect in this field, and I think it explains a lot. That in the preclinical arena, they're using single molecule high potency agonists, which are very dangerous compounds in humans, but so that you wouldn't develop them for drugs. And the clinical trials are essentially using cannabis or extracts of it. So there's a big disconnect. It's like sort of comparing opium with fentanyl. The bottom line is there is no efficacy in the clinical trials. One exception is down here on the second one. It's an area where we happen to have a patent. It's palmethoethanolamide. It's not really a cannabinoid. It interacts with CB2 receptors in some unknown way. Nada, there's nothing there. And we looked at it for neuropathic pain studies. We brought it right up to date for the NUPSIG review. It's tiny, the effect size, absolutely minuscule, if there's any effect. So in other words, there's a massive disconnect between what we know about the cannabis field, and I believe, rather like the opioid study, that the mechanisms are there, the receptors are in the right place, that they should be able to interfere with pain and nociception, but we haven't cracked it. And I think a lot of the blame can be laid at where we go into animal models. And as I'm talking about animal models, and this is a sort of fairly conventional description of the drug development pathway. And I'm talking about that stage just before clinical development. And one of the problems is we use animal models to justify going into the clinical. We like to regard them as proof of efficacy. And to me, that's too broad a question. It's not a focused enough question. We need to, and I absolutely believe these models have utility, but we need to be smarter and more rigorous at the way we use them. And we wrote about this with Jim Eisnack just a couple of years ago. The second half of the title is a well-known quote from Einstein. I said it to Jim as a joke, and suddenly it ends up in the title. But you know me, I'm not provocative, am I, Per, really? But what I want to focus on is solutions. Chris, you'll appreciate this is from Africa. So a paper that everybody should read, and sadly, what are we, 31 years later, it is still relevant, and it applies to all medical research. Is the late, great Doug Altman's paper. And I would also advocate you should read John Iodini's paper, Why Most Research Findings Are False. But the scandal of poor medical research. So this is not a unique problem to pain. And it is as true today as the day he wrote it. But one of his summaries I really like. We need less research. There's too much bad research out there. We need better research, and we need research done for the right reasons. And that really summarizes very succinctly in what I'm going to be talking about for the next 3 hours. And we've recently, I've been part of a team led by Neil O'Connell at Brunel, funded by the Europe Intranet, because this is a problem across the whole spectrum of pain research, right from molecular biology through to clinical psychology and epidemiology. People aren't trusting pain research anymore because for many reasons. So Neil led this group of researchers, and we have published this large collection of materials trying to persuade people that we need to take action and improve the trustworthiness of pain research. And ISP and EFIC have both adopted it as form of policy, and I'm delighted that the Australian Pain Society have. Next month, nearly all the main pain journal editors have come together and have adopted it for their journals. So the point of putting this up is that we're offering solutions within this, and this is a more generic problem. I'm really using just animal models to illustrate points here where we need to up our game. And there are several facets to it: equity, diversity, and inclusivity. Is that relevant to preclinical research? People think, well, not really. Absolutely it is, because nearly all preclinical research is done on male adolescents. And if you think, many of us are proud owners of male adolescents that have been some time now, but the fact that they might reflect norms of society is quite disturbing. They become normal after all. But most preclinical research, for quite good reasons, PPI, or PPE is now, Roger referred to it. Oh, this isn't relevant to preclinical. Absolutely it is. Preclinical researchers need to interact with people with lived experience to understand the problems. And that's always disappointed me, how few people involved in this area want to come to clinics. I'm talking about mainly methodological rigor and data authenticity, openness and transparency, and balanced communications, all areas that I'll top on. But this is just to emphasize, this is broader than just preclinical pain research. And who are the stakeholders? Well, researchers, and there probably won't be too many preclinical researchers here, but it's part of our area, but clinical researchers too, because you're using this information to do your clinical studies. Institutions, journals, funders, and investors. Those of us who have worked in the space of spinning out university assets to pharmaceutical companies, some people get surprised when pharma take a molecule and from some Nature paper, and they come back a few months later and say, oh no, we're not really interested. The truth is they tried to replicate it and they can't. They don't publish that. But the consumers of research, so industry consuming academic-driven research is really important. People living with pain, if there's not new drugs coming through the clinicians or people living with pain, clinicians and policymakers. This is everybody's problem to get this right. Because if we don't, as a field, then other people will do it for us. People will stop investing in pain discovery work. So back to the animals. This is Roland Rat. He's actually from my lab. He's doing something called burrowing, the apparatus. I don't mind mentioning the company that supplied it. They're called B&Q. It's a piece of old guttering with 2 screws underneath it. So we can divide this up, and we've written about this, I think, since 2008. It was our first paper looking at this, into 4 main areas. And one of the reasons I started writing about this was I was living a schizophrenic existence, or double existence. I was doing clinical research and I was doing preclinical research, and there were two different worlds about the kind of way we designed and conducted our studies. But there are four areas: external validity, which is basically about the model and the outcome measures you use to infer the presence or absence of pain. And too many people confound those. They talk about animal models in a generic sense, and you don't know whether they're talking about the symptoms or the signs. Internal validity, which is rigor, and then reproducibility and transparency. And I want to say what we're not talking about here. So this is a book we published coming out of a European network. I'll tell you a little bit more about in a minute. But we drew the parallel between types of clinical study and types of preclinical studies. At one end, you have the case report, which in the preclinical area, essentially exploratory observations. And at the other end, you have clinical trials and meta-analyses. And essentially, a lot of animal model work is a clinical trial in another species. What we don't want to destroy, or not destroy, but attack, is genuine, smart science, people following their intuition, getting hunches, following their nose, and making brilliant discoveries. But those are hypothesis generating, but the problem is they're often reported as hypothesis testing studies. And we really think you need to make the distinction, because you can't do hypothesis generating studies are the same as case reports. They're valuable, but they're not the end of the story. And these have to be replicated. And replication in preclinical science is a dirty word. It's not sexy. It really is important. If you don't replicate it, then it's not that valuable. So we're talking really about the center part of this, which is the transition between hypothesis generating studies and hypothesis testing studies. The problem is that the hypothesis generating studies are often reported as if they were hypothesis testing studies using p-values and that kind of thing. And then we'll say a little bit about replication. One thing I'm not going to talk about is conceptual replication, but it is really important. And that's when you see the really high quality preclinical papers are basically triangulating onto the problem using many different techniques: electrophysiology, molecular biology, behavior. And when they all come to the same conclusion, then you have more confidence in those conclusions. And a really important bunch of papers were published by Doug Altman and others 10 years ago, 10 years ago now, in The Lancet. They originally called it research waste, and I think that's a pejorative term. It implies that there's nothing we can do about it. So we like to talk about research efficiency. And again, this is a must-read 3 or 4 papers. They set out 4 columns. Are the questions relevant to the users of research? Are the methods used rigorous, appropriate design, conduct, and analysis? Number 4 and 5 are about reporting. So this issue of not reporting so-called negative studies, which I'll come on to again. And then in the middle, they put one in later on, which is something that every researcher will recognize: the real cost of unnecessary and over-regulation of research. And I would advise everybody to read this. Doesn't matter what area of medical research you're in. So let's look at this one in terms of animal models and see where we can go with it. Are important outcomes assessed? So what we're talking here is about external validity, and in particular the model. This is about replicating the disease. By the way, I don't believe you can have such a thing as a model of pain. Pain is a symptom. You can have a model of a disease. So you can create a model of osteoarthritis, or you can create a model of traumatic nerve injury, but you can't create a model of pain because it's a symptom. So this is a very old analysis we did, but it's still relevant. So we looked at over 4,000 animal model reports, and hardly surprisingly, 70% of those are in models of traumatic nerve injury. And there's no end to the ingenuity of people in my field, different ways we can injure the sciatic nerve and then call it our model. No names mentioned. If you go, this was the original Lancet Neurology 2015 analysis. Over 50% of the trials were done in either diabetic neuropathy or postherpetic neuralgia. And I'll come on to why that might be important. But in other words, we're doing the clinical trials in animals and humans in completely different conditions. That doesn't make any sense to me. Does this matter? Well, there was a lot of hype around microglia. It's kind of disappeared now. Guess what? we were quite concerned about this because most of the methods used to demonstrate the microglial response to, say, nerve injury were histochemical in nature, which are useful from the anatomy point of view, but they're not quantitative. So I think we were one of the first people to use flow cytometry, which is a fairly useful quantification method of cells. And we compared a whole load of different animal models. And guess what? You got a massive microglial response in a model of traumatic nerve injury. You didn't in models of HIV neuropathy, chemotherapy, or drug-induced neuropathy, or varicella zoster infection. Yet, and therefore it's a response to injury and not necessarily a response generically to pain. But where were the studies, clinical studies being done? They were being done in postherpetic neuralgia. Doesn't make any sense. What about the outcome measures that we use? Now, we can't measure pain in humans. We certainly can't measure pain in non-sentient or non-communicative rodents, but we can imply its presence or not. And we, again, wrote an article a while ago talking about sensory profiling animal models. It was really meant to be a challenge to the field. It upset one or two people, which is always good. So I think you probably all know that generally the pain outcome measure is prodding a rodent with a stick or a piece of hot something or other and looking for limb withdrawal. That's really an example of measuring what we can measure and not what we should be measuring, because we don't use that as an outcome measure in clinical trials. So we posited that these models don't have much, or these outcome measures don't have much utility in predicting pain. In other words, we're measuring the wrong thing. They might have utility as profiling biomarkers, because if you think about it, the few successful drugs that have been through have been in conditions where animals have hypersensitivity, particularly in inflammatory things, which is what you're modeling here. And of course, this goes back to Professor Lindblom. He was the real pioneer of sensory profiling in humans. So now the question is, going back to precision medicine, can we predict who responds to which drug on the basis of their sensory profile? But then we should be conducting the clinical trials appropriately. But then that leaves us high and dry. How do we assess whether the animal is in pain or not, if we've taken away the main way of dealing with it? And just to go back, so this really, you can date all this back to Ulf right at the beginning. But this was a study that many of us were authors on. Our German colleagues put together a whole data from across the world. And essentially in neuropathic pain, except interestingly in leprosy, which is one of the areas we studied, you can, to put it extremely crudely, separate patients irrespective of their underlying disease into one of three sensory categories. But certain diseases skew more to one. So diabetic neuropathy, you'd expect them to be in cluster 1. Cluster 2 is where you have preserved sensory function, but heat or cold hyperalgesia. And the only one that is close to what we do in animal models is mechanical hyperalgesia. And that may be why postherpetic neuralgia, because there are these patients in the postherpetic neuralgia groups, is a relatively good clinical trial model. But if we're doing diabetic neuropathy studies, they're not the same profiles we're studying in rodents. So I had a conversation with a colleague who's a field biologist, and he looked at me rather as if I was mad, which many people do, and said, hang on, you're studying pain in rodents. Why would you want to do that? Rodents are a prey species. They wouldn't display pain behaviors because that would identify them as prey. And this is where you have to separate rats and mice. Anybody who works in a lab knows you're much more likely to get bitten by a mouse than you are a rat. Mice are solitary, usually quite aggressive. Rats are actually quite social creatures. They live in these complex burrow systems with all ages and both sexes, and they build these burrows to shelter from predators and that kind of. So once you start to think of the behaviors you're likely to see as predator avoidance behavior, and this goes back to an absolutely fantastic book. You can buy it for about £9 on Amazon. Barnett's book on the study of behavior of a rat. And Pat Wool, who was one of my mentors, had a rat living in his office for a while. Well, he probably did, because it was quite an old building. He probably had more rats than he realized. To observe its behavior. And Barnett wrote this by talking to people like gamekeepers and farmers and rat catchers who really understood the behaviors of these animals. So in other words, it's the biopsychosocial model of disease. And Alex Koselnik, who's a really great behavior animal ecologist in Oxford, also points out the dangers of anthropomorphizing human emotions to rodents. I get very uncomfortable when people talk about depression and anxiety in rodents. Yeah, you can model concepts around that, but not the actual condition. You know, I get depressed because Trump's in the White House, not well, we did sell the ISP office, which is 400 yards from the White House. We weren't too keen on the new neighbours, you know. Actually, we sold it just before he came into office, but we'd like to take the credit. So I'm not claiming by any means these are the behaviours you should study, but they're the ones we happen to study. And the first one we did was a behaviour called phygmotaxis. Any of you who've seen Tom and Jerry cartoons will recognise this. Rats or mice will stick to the edges of arena. It's a paradigm used in various guises in psychiatry research, and because that's the safe space from the predator. But every so often it'll go into the middle of the arena to see if there's any food, or there might be a lady rat in there, who you never know. And we were able to show, and this is a systematic review, just an overview, that that when you suppose the animal is in pain, it will have increased phygmotaxis. In other words, it'll stick to the walls. Now, the other problem is people talk about pain behaviors. Now, these are just behaviors associated with an animal being stressed or not well. So you have to contextualize it to pain. And the other one was burrowing. As I said, it works in rats, but not mice, or shouldn't work in mice. It actually does. And it's incredible that this behavior is preserved through many generations of breeding laboratory animals. And basically, rats, when they're well, burrow to maintain their burrows. Again, this is where the concept of using teenage boys as the species, because any of you know that teenage boys are not particularly keen on housekeeping, but there you go. So we could quite easily, and I have to say, those first two aspects, many smart people are working on. They're improving the animal models to make them look more like diseases, and they're improving the outcome measures. And there's a huge number of really smart people working on that. I've now closed my animal lab, but the second thing is, and this is for the clinical area, that 50% of studies, 99.9% of preclinical studies are made without reference to the pre-existing literature in a systematic manner. And most of you will be familiar with systematic reviews. Again, I had the privilege of being in Oxford when Andrew Moore and Henry McQuaid started in the pain field, and Andrew Sackett was there more generally. It's very exciting. They're very new to the clinical field, but they're actually incredibly useful, but they're tough to do. We've done quite a few now. And Nadia Solomon, who until recently was in my lab, she's now moved to UCL, is probably one of the people who've really pioneered this. And I'm not going to read these because you're familiar with them. They're the basic concepts of why we would do systematic reviews and meta-analyses. And we've done a few now. And again, this is the process of it. You have a protocol which you register, you search various databases, you deduplicate, select your studies. If appropriate, you'll do a meta-analysis, and you should always publish it in some format. But this is a really new, and you can probably just see it here. Our friends in Edinburgh, Malcolm MacLeod and Emily Senna, have a platform for learning about preclinical systematic reviews and actually doing them, a bit like Cochrane for preclinical work, and it's brilliant. It's called SURF. And guess what? Their tutorial system is called Learn to Surf, but I'm not responsible for that. So we looked at chemotherapy-induced neuropathy, and we did our initial search in 2012, just after the Olympics finished, and we came up with 180 studies. But in the time we did that, that took 18 months by all the screening. By the time we'd finished that, we did another search, and the number of publications had doubled. So that's a unique problem to most clinical systematic reviews are relevant the day you finish the search. but it's not too much of a problem because only a few studies accumulate. There's a massive accumulation here. So, and this is the one time in my life when we were ahead of technology. We used AI methods, text mining and machine learning, to extract the second phase of this. And we were able to show, and we think this is useful data for people who want to use these models, which chemotherapeutic agents give you the most sensory changes, so paclitaxel and vincristine and oxaliplatin, which you'd probably predict from the clinical arena. These are the outcome measures that are used. Now, there's very few at this time using complex behaviors that I talked about, so these are the limb withdrawal. But here's the thing. 11,500 of those subjects were male mice and 1,700 were females. Yet these are chemotherapeutic agents that have been used in breast and ovarian cancer. So there's a big disconnect there. I'm also really, really proud of this paper. I've got a couple of these, and that the first author is called Gillian Curry. So it's curry and rice. And if you come to my war history talk, which is in a couple of days' time, we're talking about sensory neuropathies in Far East prisoners of war, which were cured by Marmite, by the way. Then they had Marmite as prisoners of war. Gillian is also an author on one of those, so I get that in as well. And of course, the reason they got neuropathies was because they had a diet that consisted mainly of polished rice. And we can also show from this which pharmacological interventions respond to that model. And that's really useful information. You can see that it's actually morphine and gabapentin, and that's because they're used as the control, internal control quite often. And that's information because the problem with gabapentin in these, that people often use massive doses, 90 milligrams per kilogram, which are basically anesthetic. But you can tell from this which works. The other thing we can model, and I won't go bore you with the statistical details, is trim and fill analysis to detect something, or probably detect something called publication bias. Because a massive problem, particularly in the preclinical literature, but also in the clinical literature, is people only report studies where their hypothesis is proven. And that meant we were missing about a third of the literature, the way we modeled. And most of that literature is on the negative side. So you're overestimating efficacy. And one of the things that, there's a few things that make me lose my cool in a lab meeting, one of which is someone talks about negative or positive studies. There is no such thing as a negative or a positive study. There is a really poor quality study that's not asking an important question, and there's a high quality study asking an important question, which is relevant irrespective of whether the outcome is proven or not. So a colleague of mine does a huge number of trials in intensive care. Most of them have turned out to be negative, but they're really important because they've guided treatment and stopped people using dangerous or ineffective treatment. This is the easiest part of this. This is about rigor, not taking adequate steps to reduce biases or inadequate statistical methods. We're talking about internal validity. And one thing to remember, this applies not only to the design and conduct, but also to the analysis and reporting stages of a study. And we like to call this the elephant in the room, because it is the easiest problem to fix and also the most difficult, because it requires a culture change, particularly away from this concept of positive studies only being reported. This could be instituted tomorrow, and in fact, they have in stroke. I probably don't need to tell you any of this, but we all know what the risks of bias are. They're more high quality studies with low risk of bias are more likely to yield accurate answers. Biases can work in both directions, and some are trivial and don't matter, and others are substantial. And it influences all stages of the pathway. People often think it's only about blinding in your actual conduct. No, it's about how you design your study, how you analyze. So people tell you if the observers are blind, do they tell you whether the analyzers were blind as well, and how you report it? And this came from really three colleagues. I've mentioned Malcolm McLeod. He, like me, he was a stroke physician, I was pain, but we were doing clinical and preclinical work and completely disconnected. He was working in stroke. Emily Senna was one of his PhD students, and really rejoiced that she recently became a full professor at Edinburgh University. Malcolm's at Edinburgh. And David Howells, I recently had the pleasure of visiting again in Melbourne. He's recently retired as a stroke researcher. It's one of those coincidences of history. We're both keen trout fishermen. He grew up in Aberystwyth, and I discovered when we were both about 12 years old, we were fishing the same river near Aberystwyth at the same time. Problem was I was fishing with his headmaster, who was a friend of my dad's, so he didn't remember that quite so well. Now, they came up with what they called good laboratory practice and tried to identify the main areas of risk of bias. But the problem with these it's not necessarily a problem, but there's assumption because they're the same risk of bias we identify in clinical trials. And then they may not be so important for animal models, and there may be other causes of bias that we don't know about. So this is the Rumsfeld known unknowns. But what they were able to show is that the more of these items of risk of bias were reported, the effect size gradually incrementally goes down. So in other words, the more rigorous your study, the smaller the effect size, so it eventually vanishes. And we've done quite a few meta-analyses, big meta-analyses in pain research for interventions like cannabinoids, for models like chemotherapy, and for burrowing and thigmotaxis, and we're also just reporting grimace. Only 50% of studies report a blinded outcome. They don't tell you, and they don't give you enough detail to decide whether the blinding method was robust. Only 8% report allocation concealment. That's when the person creating the nerve lesion, for example, knows the allocation of the group. 50% report randomization, but if you drill into it, most of them don't actually randomize at all. They might be the order in which they take the animals out of the cage. I'll tell you about animal exclusions. 15% report a sample size calculation. So you're doing your experiment without a sample size calculation. That is bizarre. And the reason is that if you look at sample sizes, and nearly all preclinical, they're exactly the same. They've gone from 8 to 6 during my career. The reason is that the number of rats you're allowed to keep in a cage for welfare reasons went down from 8 to 6. And these is something we came up with in the Entrust PE, and it applies across all areas of research. We call them questionable research practices. We call them other things in the pub, but and these are design, analytic, or reporting practice that have been questioned because of the practice to be employed for the purposes of presenting biased evidence to support an association. I've talked about Andrew Moore and Henry McQuay when I was in Oxford. I was also taught by quite an eccentric clinician who you'll remember well, called Chris Glynn. He came up with many Australian sayings, some of which are repeatable. He also used to write in the notes in turquoise ink because he could always find his place in the notes. But he said, never trust a scientist who believes their hypothesis. And you should, from the moment you make your hypothesis, mistrust it. So underpowered experiments. P-hacking is when you don't have a statistical analysis plan, you just run loads of statistical tests until you find out one that gives you the result you want. Inappropriate data redaction. This is a big problem in preclinical research. If you look at the numbers in a report, you'll see that they vary, often within the same paper. And that is because people are taking out so-called outliers. And I'll show you why that's dangerous. And some very distinguished pain researchers had the courage to actually stand up in front of the funders and say they do that routinely and they don't see why they should report it, let alone having pre-established criteria. And then hypothesis after the results are known, or harking. And then presenting post hoc analyses, which are fine, but present them as the primary outcome. And the incentives for this are diverse, ranging from naivety to trusting your own hypothesis or believing it. And we have a vested interest here. Actually, if you look at industry, they want to know which things don't work early on. So they apply many of these things earlier on, but they don't publish as much. That's one of the problems. And you know, it's a well-intentioned confidence in the veracity of your hypothesis. It must be right. I heard a brilliant comment at the Australian Pain Society weekly. We just need for the evidence to catch up. That was said in a really odd session about psychedelics presented by a lady who was a psychologist who had clearly a big practice in this area, didn't think it was worth mentioning that as a conflict of interest. And she had the most wonderful job title, which was she's a psychedelic assisted psychotherapist. So I'm now an evidence assisted physician. This is an example of why taking outliers out is so very dangerous. It comes from Uli Dernagel in Charité in Berlin. He's one of our collaborators. He's a stroke researcher. And he showed that the kind of numbers you'll see on the x-axis here, you see in standard animal experiments. If you randomly take animals out, you just reduce your power. But if you take out the outliers, then you massively increase your effect size. And it's fine to exclude animals die and exclude them, it's fine, but report you exclude them and why you exclude them and have reasons before you start. If you did that in a clinical trial, you'd lose your registration as a physician. And we were really lucky, we got some funding from the EU to work with a whole lot of different colleagues across Europe in something called Equipped. And we produced some tools that I hope are important. And one of them was we published in Nature Methods a couple of years ago, a framework for rigor in the design, conduct and documentation of animal experiments. And we break these down into 5 domains, design, conduct, or 5 areas in different domains. Now, Jan Vollert was in my lab then, he's now got a position in Exeter. He said to me, we do need to do this from a narrative. And I said, no, we need tick boxes. And this is where I publicly admit that Jan was absolutely right. This is a narrative thing. Because the problem with tick boxes is they don't make people think. These, hopefully, we pose the right questions. So you must have a predefined hypothesis, and but it's fine to do hypothesis generating experiments. Just tell us that's what you're doing. Don't report them as hypothesis testing experiments. And the sample size needs to be, and we say the default position is, if you can't prove it's a hypothesis testing experiment, then it's a hypothesis generating experiment and should be reported as such. Plan your methods analysis, and if you're doing a hypothesis testing experiment, register your protocol like we would for a clinical trial. And there are plenty of good registries for these at the moment, and obviously include appropriate controls. Most of us are terrible at statistics. I am, but you need a statistician. If you're doing an absolutely pivotal clinical trial in an animal model, you need a statistician, and you have to have a statistical analysis plan. That might seem obvious, but it prevents p-hacking. And I don't need to sort of tell you about this, but not only should you randomize a blind, but you should do it appropriately. Now, the key point of this is right at the end, we say, we know you can't do all this. You might be working on a transgenic mice where the wild type is white and the transgenic is red. You clearly can't blind that experiment, but just tell us that you can't blind it and why you didn't. And we produced a number of other data. We produced a book, and Kim Weaver in Nijmegen in the Netherlands led a fantastic e-learning thing. And we, in parallel to this, have a quality system for labs, particularly academic labs who want to sell their patents onto industry. And we have a quality system. And there are others like the NC3Rs in the UK have an absolutely fantastic electronic online system that is free to use. The last main area I'm going to talk about is inadequate replication. So replication is the key to good science, independent replication. But people just don't do it. I got really frustrated when I would sit on grant funding committees. People would pompously say, well, this is merely replication. It's not novel. It's not new. No, but it's important. And this is about reproducibility. And we've sort of come up with a framework. I'd love to say we set out to do this framework, but we didn't, but it's kind of emerged over the years. So I'm not going to talk about the first one, but in the first trial of burrowing, we did it independently at Pfizer and at Imperial College and reported them. Now, we didn't use the same methods, but they came to the same conclusion. But I wanted to talk about prospective multi-center animal studies. And this was led by Emily. It's our first sort of big grant funded by the EU. It's called Multipart. A lot of the Europeans didn't understand why we thought Multipart was so funny, because it's an automotive manufacturing thing in the UK, but it apparently doesn't exist in Europe, so nobody understood what we were talking about. And we know the methods are well established, and we suggest that they could be used for 2 reasons. A, replicating a new model or outcome measure, and I'll give you an example from burrowing. But really, and it's really odd that we don't do this, is replicating the key molecule or ion channel or that has just been reported in an Armani journal like Nature, replicate that. And Nature are beginning to ask for replication studies, but not multi-center ones. They're really easy to do. The study I'm about to show you only took 6 months and cost each lab very little. But you can replicate, you can generalize. There may be issues that are unique to one lab. You can standardize rigor, but also you get really good feedback from the people who are actually doing it in the other labs, the technicians and people saying, actually, you could do it this way and it tends to work a bit better. So there's useful parts to it. And we did this, as I say, it only took us 6 months. Rachel Wodarski, who I think is now, I think she's in a place called Manchester. Is that near here? Okay. And we did it in some academic and industry labs, mainly in Europe, but also in Japan and the US. And we took complete Freund's adjuvant injection into paw because we're fairly certain that that is an animal model that where pain occurs because someone injected themselves by accident a few years ago and wrote it up and said it's very painful. And we designed a protocol, etc. We had a standard protocol that's available for anybody to see. Bottom line here is in red, you can see that there's decreased burrowing behavior, not necessarily due to pain, but probably in this case, that writes itself after about 4 days, which is about right. And we don't see that in the black line in the sham animals. And importantly, we don't see it in the naive animals. And experiments doing complex behaviors should always have a naive group because you don't know what's going on in the animal lab. You don't know whether the technician has been digging up their onions that weekend and all that, which happened to us. Or everything went off and it was because he was smelling of onions. So that green group is really critical to show you if there's stability in the lab and the assay. Of course, not all centers got the same result. And here some of my basic science colleagues threw their hands up and said, this is all rubbish. I'm not going to mention who they are, Jeff. Because you can't get the same level. If you saw a clinical trial where every center was getting the same outcome, you'd be very, very suspicious. But most centers did. One center in Manchester didn't, but that's fine. And we were able to look at that center, and they were very new to using the technique, and therefore we were able to identify where the source of error. But we could reproduce it. Tragically, that remains the only, we think, the only multi-center study published in pain research using animals, and it's easy to do. And if I was wanting to flog something to industry, that's the first thing I would do. And I'm surprised industry don't ask for it more often. There was another one which I was involved in the US, which took many of the big labs in the US, really low-hanging fruit, which is just the response to a von Frey filament in a nerve injury model. We're now 6 years down the line and they haven't reported it yet. The bottom line is they couldn't replicate it between labs. That's the standard outcome measure. And we also feel, and I would just like to suggest to you, that a third stage, that after a while, sufficient data has accumulated to perform a systematic review. And this is just one, and it shows the advantage of registering your protocol, because we discovered that Anne-Marie Hagard in Copenhagen was doing the same thing. So we just said, well, let's get together and do it together. But this, we looked at burrowing. We've also done it for Thigmotactis and other things. It's also a really useful training tool for young scientists, particularly those in remote locations, to 45 studies, over 3,000 rodents, 33 models, 27% different drug interactions. It's amazing how many of those drugs you never see again after that. But mainly gabapentinoids, NSAIDs, and opioids, because they're used as the internal controls often. Bizarrely, it's reported in mice, because mice are not fossorial rodents, but again, male only. The reporting quality is improving a bit. If I'm really arrogant, I would say some of our stuff comes from our lab where we like to think we had high standards. And there's some risk of uncertainty. But the main ones that I worry about are the animal exclusions not being reported. And there are complexes in this thing because some animals are not innate burrowers and you have to have methods of training and things. It's not as simple as it sounds. But we were able to show that overall, and actually there's little evidence of publication bias here, negative studies not being reported, animals that you would hypothesize to be in pain burrow less, and it doesn't really matter what substrate you use. Sprague-Dawleys are slightly better at doing it than Wistars. Somatic injury models as well as nerve injury model both have much the same effect. And you get a response to some, but not all pharmacological interventions, which is what it should be. So I'm going to, so I've really brushed over an area here, but I think it's a really valuable resource to create for other researchers, where they, if they want to do a chemotherapy-induced neuropathy model, or they want to use burrowing or thickening, they can go to a massive systematic review and do their sample size calculations. I'm going to slightly gloss over this last area. It's very common in clinical practice, but it relates to, are all studies reported? No, so-called negative studies are not generally reported. And are those reports unbiased? So we have something called the ARRIVE guidelines, which many of you will be familiar with, CONSORT for clinical trials. They really revolutionized how we do clinical trials. These are the guidelines for reporting animal studies. They were updated 5 years ago. Many journals have adopted them, but how many police or really institute it is another question. And the PAIN have not. The other problem is conferences. You get 7 minutes to give a free presentation, or someone drops by your poster. I would argue I want to know more about the methods first, but other people, want to see the results. And so Shai Silverberg has come up with this so that authors don't have to go through all their rigorous methods, because that would take up your 7 minutes, but you can use these icons. Now, they do require knowledge and honesty, but we're going to try them out at the World Congress. And these were developed by my great friend Shai Silverberg, who is, has got a wonderful title, the Director of Office of Research Integrity at the National Institute of Health. It's only second best. I have nearly the best professorial title, Imperial Professor of Pain, but we do have a Professor of Control and Power. Believe it, wouldn't you like, straight out of Harry Potter? And the last thing is about spin. And I told you before that I was educated by Henry and Andrew and people early on that you read the methods first and then decide the paper is worth it. Because it is surprising how often the spin, that title, the abstract, the conclusion doesn't match the results. And that was particularly problematic when we did the cannabinoid stuff. So Andrew and Chris Eccleston, who I think they've both been lecturers here, came up with this fairly simple tool for detecting spin. And then that level of spin gets even worse when you go to the mainstream media reporting this fantastic finding, getting patients' hopes up, and they spin it out of all control as well. But spin or narrative bias is really important. And the last thing we decided to do, and this is when my colleagues finally thought I'd lost my marbles, is many of these behaviors like Figma-Tactus are recording using videos. So why keep those videos secret? You should actually publish them when you publish your paper and allow other people to look at them. This is one of the few times this has actually been done. We kept the videos blinded so we know who's doing it, and then we give them the blinding code when they finish their study. So this is where I'm going to end up. I've talked about potential solutions, but I'm going to talk about the easiest but the most difficult. And also, I'm very conscious that I've created a research career, a bit checkered, but I've created it on the basis of quite an easy world where we didn't have to do a lot of this. And most postdocs and PhD students absolutely get this, but they also understand the system that they have to publish Armani papers to get the grants, to get the, and they're not, who's going to do replication studies, to get the grants, to get the tenure position, to get the promotion. And I really feel for them. So it is about changing institutional culture. And that everybody is, everybody, the funders, the journals, the institutions. Those of you academics will know we have something called what's it called now? The REF, the Research Excellence Framework, where academics are assessed every 7 years, I think, something like that. I'm going to retire just before the next one. So. And we've managed to get some of this into the REF. But at the moment, the problem is you're rewarded for novelty. Even if that novelty is not reproduced, even if that novelty never ends up in a drug. It's a bit like the city giving share options too early on. You should be assessed on your long term. So in many big companies, the preclinical people were rewarded for delivering a compound to a clinical trial. No, you should actually be rewarded as to whether that trial worked or not. So what we want to tip the balance for is where people are rewarded for doing rigorously designed, conducted, analyzed, and reported studies ask important questions. We need to ask the people with lived experience what's important, and report on the findings irrespective of the outcome. And finally, your findings should be reproducible. And if we can change that culture, but it also requires us to expect failure and celebrate success. Whereas at the moment, we have far too many successes that turn out to be failures. And as I said, this is not unique to pain research. One of the privileges of being president of IASP is you get to choose a few presidential task force. I'm doing a really exciting one with Roger on environmental issues, both global warming, but also something that we were unaware of is the major role of environmental damage from analgesic pollution of water systems, which turns out to be a really interesting and hot topic. But this is, we're revising our laboratory animal guidelines. Many of what I've talked about with this, Anne-Marie and Esther are leading it. Manfred Zimmermann wrote these in 1983. They were a very highly cited paper from Pain. I suspect cited more often than they were read. But that dropped off. And of course, after 40 years, they're irrelevant. So we will be publishing ISP's new guidelines probably sometime early next year. And you'll hear about them if you come to Bangkok for the World Congress. I'm really excited. We're going to Asia. That view of the Royal Palace is just opposite, on the other side of the Chao Phraya River, Siriraj Hospital, which is a fantastic institution where ISP have our clinical training fellowships for Asia that have been immensely successful. And that will be in late October of next year. And just to end up, just by leaving this on the screen, there is tons in the Entrust PE about trustworthiness of pain research. This is not just about animal models, it's about all the research we do. And there are really, really good materials, but it's going to require some cultural change. But and we give a lot of tools in there, but it's been really encouraging is how many organizations are taking it up. And I say most of the journals in the field recognize that we have to up our game here. Thank you, Chris, for inviting me. Thank you for remembering Ulf. I've been Andrew Rice. Thank you.
Thank you so much, Andrew. Any questions, comments?
So what do we do with all this rubbish research published? Can it be kind of cleaned up and that new researchers don't need to go through all this hoops again? And
it's a really difficult question. I don't know. I mean, we certainly should be doing less research and better research. And one of the problems in the systematic reviews is we include all of this, despite that a lot of it is low quality. I guess the hope is we reach a certain point, as we did with clinical trials. I mean, we all remember how they were revolutionized by Cochrane, by pointing out that most were bad and what you needed to do a good trial. So I hope it's more a question of education, a question of how far you go back. And sometimes when I give this talk, I stick up studies of some of the early slides of the early studies I did. I don't know what we do about that, but I hope what will happen is the quality will change. People will see the advantages of quality, and then we can do much better systematic reviews of meta-analysis. It's not a direct answer to your question, but there's a lot that needs to be discarded. But this is not unique to preclinical research, and it's not unique to pain. There is tremendous pressure on people to publish, and that is problematic in my view. In certain countries, your salary gets an enormous boost if you publish in an Almani journal. That is not a good incentive.
Do you think we also need to be thinking about the way that we do peer review in terms of the journals request peer review, but how our systems of peer review don't necessarily feed into this?
So the biggest headache for journal, well, there's a couple of problems here. Journal editors have a lot of things that keep them awake at night. The biggest one is paper mills and AI created fictitious papers. And if you talk to any journal editor, that really keeps them awake at night. It's a big worry. But peer review is really difficult. Most journal editors really struggle to find peer reviewers. and we don't have the time, we're not rewarded for it. So one solution is to do less research and have, that would reduce the peer review burden. But some journals have adopted some quite interesting approaches. So the Lancet and I think it's Anesthesiology have a professional statistical reviewer who actually sees that before it goes off to the academic peer reviewers. And it might get rejected at that stage because many of the ones that I wasn't certain about, I wasn't clear about the statistics. I had a sort of rule of thumb: if they're using a statistical test I've never heard of, then there's something fishy here. But I was not an expert in that. So that's one solution, is to have that stopgap of thing. But it's a difficult problem. But on the other side of the equation, it's a bit like Churchill and democracy. It's a very bad system, peer review, but it beats all others that have been tried to date.
Yeah, I agree. But I think when maybe in ref and those sorts of things, that as academics, there should be some level of reward for the peer review that you do, because it's not factored in terms of good academic practice, is it? No,
it's the kind of thing that probably should be rewarded in appraisals and things like that. I mean, there is a kind of incentive that if you do enough peer reviews, you often get onto the editorial board and being on it. No, I'm not being facetious. If the editor sees someone doing really good peer reviews, and they're often the younger researchers, then they'll eventually get onto the editorial board, and being on the editorial board is really important for career promotion. But the ref has also moved, so we have impact statements that look at things over a longer time. That is really important, and there's less emphasis now just on your 3 best papers and that kind of thing. Oh, and actually, what I can tell you, I'll come to. Malcolm did a really scurrilous thing. He looked at the outputs from the 5 biggest universities, that include Edinburgh and Imperial, from the last but one ref for all their animal studies, and saw how many met the rigor criteria. Yeah.
Yeah, I just wanted to mention a different BPS, a British Pharmacological
Yep.
Society, and our journal. So some years ago we decided to be much harder line in terms of editorial policy, and so every prospective author now has to tick all the rigor boxes that you showed, Andrew. And of course, what happens is submissions go down because you can do crappy studies and publish them somewhere else.
But you get better. I mean, what I will say here is,
for sure,
hats off to the BJP, because they are one of the journals that have really led the charge on this. They were there from the outset. They really prompted a rise. They've been fantastic. But I'm not sure it's a bad thing that your number of submissions goes down, because your peer reviewers won't be seeing so much rubbish, and hopefully you're getting better quality stuff.
No, it's just the crap you get from Wiley saying, why have your submissions gone?
Oh, okay.
Because we're publishing.
Because the publishers, absolutely, yeah. So it's difficult, but really hats off to the BJP, because you've led the charge there. As have the BJA, the British Journal of Anaesthesia. They've done quite a lot of good things as well.
I mean, you just touched on it just now, but can you just give us some idea of how you're dealing with the scourge of AI generated papers?
I'm dealing in a very simple way. I'm not a journal editor. That is not entirely facetious. It's getting, I mean, we see it at 2 levels. One is student submissions for BSc write-ups and things. So universities are really worried about this, and they have got systems, but it's a cat and, it's a catching your tail thing, because the systems get better and you get better at detecting them. So it's problematic for universities. It's problematic for editors. One suspects that sometimes things get through that shouldn't, because they're pretty sophisticated now. But it's a headache, and there's no solution, because it's evolving the whole time. And
there might be a lot of retractions in the future, some people pick it up.
There might be a lot of retractions. So I haven't covered retractions, but most of the retractions we see are, except for 1 or 2 really spectacular answers, one of which led to the downfall of the chief of Stanford University, so the top university in the world. His mistake was retractions are a big issue, but it tends to happen mainly in preclinical. NUPSIG were very involved in one that should have been retracted from New England Journal of Medicine clinical trial. Some of the, and then the fraud, the difference between bad research practices and fraud can be sometimes quite a narrow thing. But one of the big problems is images, because a representative image means my best image. whether it's a Western blot, or whether it's a histological image, or whether it's a brain scan. Because that's human behavior, we're going to do that. So somewhere that have been able to detect fraud have been by looking at analysis. And several people, I think the one from Stanford was this one, where they found the same Western blot essentially in 2 or 3 different papers. The issue was we work in big multidisciplinary groups now, that the boss cannot always know that. But that particular boss, it was drawn to his attention and he did nothing. And probably somewhere in my 300 or so papers, there's something that is not quite right. I don't know. I think any researcher would admit to that we don't know everything that went into our papers, particularly if you're in groups doing different parts of the experiment. But retractions are a big issue, and I think they'll become more common in preclinical science, but detecting, and they'll be mainly around images and image fabrication. Thank you.
Thank you. Andrew, come and sit down. Thank you very much indeed.
Thank you very much, Andrew, for a very thoughtful presentation with, as you say, solutions, but I think we're all going to go away with something and think about what we are doing and the impact of what you said. I think the Entropy work was extremely interesting and brought together such a diverse group of people. I don't know if you've seen it, but actually I saw just a couple of weeks ago that NIHR have adopted it in a call for the intravenous lidocaine.
I didn't, yeah.
And I think this is really here to stay, and I would really encourage people to go away, researchers to go away and look at that. And there's so many on the website, not just the paper. There are so many resources that are useful for different those different audiences that you were describing. Thank you, Per, for your lovely. I always like hearing about history of and how individuals' careers develop and what takes them, and starting from that very basic physiology around a sensation and then looking at where a career develops. Finally, thanks to Chris and the Pain Relief Foundation for a great lecture, and I as the BPS representative, very much look forward to this collaboration. Thank you.
Thank you very much. Thanks for you guys still coming physically. Thank you for all the people that come online. And I'm going to thank Roger and just